welcome back, Bio. We are finishing off section 3 in chapter 12. This is focused on, so far we've just focused on how RNA, and then yesterday we focused on how RNA is made using DNA through the process of transcription, and now today we're going to look at how RNA makes protein through the process of translation. So translation is the decoding of mRNA message into a polypeptide chain, or simply protein. And it will take place on ribosomes. So we said ribosomes, if you remember going way back to one of the first chapters we talked about, their function was the synthesis of proteins. Now we're going to take really an in-depth look on how that actually occurs. And probably the best definition for translation, probably the easiest one to understand that gets most straight to the point, is that during translation the cell uses information from messenger RNA to produce protein. So that's what you're going to need to know for translation. And so step by step we're going to look at how translation occurs. So the first step is that messenger RNA is transcribed. Remember, that's just making RNA from DNA in the nucleus, so that's step one. And then it enters the cytoplasm where it attaches to a ribosome. So after messenger RNA is made in the nucleus, it is kind of sent out into the cytoplasm where it'll meet up with a ribosome. And so that's step one, meeting up with the ribosome. And step two is it will begin when an mRNA molecule attaches to the ribosome. Again, that's what we just said. And as each codon, which remember that's the three little either A, U, C, or G, as each codon of the mRNA molecule moves through the ribosomes, the proper amino acid is brought into the ribosome by tRNA. So remember, tRNA is transfer RNA, and its job is to transfer amino acid. So it'll bring us the proper amino acid to the ribosome whenever it is needed. And in the ribosome, the amino acid is transferred to the growing protein, or remember, polypeptide chain. Protein is what, and just another word for that. So it'll bring in and it'll transfer it to our growing protein. And then each tRNA, transfer RNA molecule, carries only one kind of amino acid. Then in addition to an amino acid, each tRNA molecule has three unpaired bases. So it'll have three A, U, C's, or G's that are kind of haven't matched up with anything. And these bases are called the anticodon, and because they will match up with the mRNA codon. So we already have our three sequences from our mRNA, and now the tRNA is going to bring us three more that will match up again to form a full RNA strand here. And then our next step is that the ribosome will bind the new tRNA molecules and amino acid as it moves along the mRNA. So here's kind of a picture of what this looks like. A little bit confusing, but it kind of just makes sense, is that our ribosome, its job is to make proteins, so it's going along this strand of mRNA that will eventually make a protein, and it's going along and it's just sticking on our new bases and our new amino acid as it goes. So it's just building the protein, that's all it's doing. As you can see there, it's continuing to build the protein. And this process will continue until the ribosome reaches a stop codon. Remember, the stop codons will signal the end. It's going to stop its whole process. And you can see up here on the top, we have our polypeptide chain, aka protein, all these different colored dots there. So eventually, it will come to a point where it stops, and this will be a protein that's ready to go serve a function somewhere else out in the cell. So that's pretty much it. So, quick review. DNA is replicated to make more DNA. So that's our first word we learned. Then DNA is transcribed into RNA. Then RNA is translated into protein. So that's the steps we need to know. It's DNA to RNA, RNA to protein. If that's all you take away from this, that's good. Then that's what you need to do, because those are the most important key ideas here. And so then our last slides for this section is just going to be some roles of RNA and DNA, which will be a little bit of review. And this first part is kind of what I just said, is that the cell uses the DNA master plan to prepare RNA blueprints. So the DNA kind of is the brain, and it makes blueprints in the form of RNA, because our DNA stays in the nucleus. It doesn't want to go out and actually make it itself, so it sends RNA to go make the proteins. And then the RNA molecules go to the protein building sites in the cytoplasm, the ribosome. So that's a pretty good analogy to use here. The cell uses the DNA master plan to prepare RNA blueprints, and the DNA will stay in the nucleus. The RNA molecules go to the protein building sites in the cytoplasm, which are our ribosomes. And the next is we're going to look at genes and protein. And genes contain the instructions for assembling proteins. And many proteins are enzymes, and enzymes, they kind of break things down, kind of get things going as well. And then their definition is which catalyze and regulate chemical reactions, which is just kind of to get chemical reactions started, basically. And then proteins are each specifically designed to build or operate a component of the living cell. So each protein has a specific job within the cell to kind of help it survive. Then our last slide for this section is that the sequence of bases in DNA is used as a template for mRNA, and the codons of mRNA specify the sequence of amino acids in a protein. So that's it for Chapter 12, Section 3. Let me know if you have any questions.